Start. Okay, hi everybody. Welcome to our COVID-19 forum presented by our esteemed science faculty, Diana McBeath, Andy Clifford, and Carrie O'Connor. I'm going to go ahead and turn it over to them. I'm going to ask, please, that you make sure your mic is muted for this point so that if there's any background noise, it's not interrupting the presentation. And if you have questions as the speakers are talking, if you can for now just type them into the chat, and then at the end of the forum we'll have time for some Q&A. With that, Diana, go for it.
So as you see here, here's a picture of this virus that is causing all of our issues at the moment, and the reason why we are actually sitting here at home instead of being at school. Notice the knobby proteins here, very, very significant, as we're going to get into in a second. Just to give a little background here, since it may have been a little while since you had biology, viruses technically are not living. We cannot classify them as living things. They are not cellular. They are below the level of the cell. So because of that, we really cannot place them in a domain or a kingdom. This virus is what we term a retrovirus. It has a RNA center, not a DNA. If you recall, those are the nucleic acids. So all a virus is a nucleic acid center with a protein coat. There's no cytoplasm, there's no nucleus. By our definition, they are not living. So something that we can't even classify as living can kill you. So as I tell my students, we are really in sort of the twilight zone here with these entities. Dr. Clifford will probably agree that perhaps we need to change our definition of what we define as life, but right as of now, these are not classified as living things. So this is, you probably have seen this all over the place, a nice three-dimensional view of this virus. Technically, you hear the term coronavirus, but technically it is SARS-CoV-2. There is a SARS-CoV-1, if you recall, caused some issues in 2008. Here is the picture that a student of mine just sent me, which I thought was kind of cool. This is a student who's really into combining science and art, and you get a nice view of the cell getting ready to attach to a cell, which is really very, very significant here. We're going to talk about that in a second. This is also quite a unique virus in that, as I mentioned, not only is it a retrovirus, which is an RNA virus, but what's also a little bit different from some of the DNA viruses is when the virus attaches to the cell, the entire receptor virus complex is taken into the cell by what's called receptor-mediated endocytosis. So the whole thing is taken into the cell. With some of our DNA viruses, just the nucleic acid enters the cell. Dr. Clifford's going to talk in a minute about how this is a unique virus in terms of its cycle. Normally, when retroviruses enter the cell, the RNA has to be changed to DNA, and then the DNA is placed into the nucleus. This virus is unique in that it doesn't do that. So Dr. Clifford will talk in a second about the uniqueness of this virus in what actually happens when it first enters the cytoplasm, which is why the infectious nature is a little bit quicker than normal. Okay? Now, since may have been a while since the whole biology days, the significance of a virus is it must have a receptor on the surface of the host cell that the virus can attach to. That's why viruses are so cell specific. The spike protein that we just took a look at in this particular virus attaches to the membrane receptor called angiotensin converting enzyme 2. Angiotensin is actually involved in blood pressure control. So Dr. Clifford probably add to that a little bit. So that's why this virus is so significant in people with blood pressure issues and heart issues and things like that. But the connection of the spike protein to the enzyme is everything. The only way that this particular virus or any virus can attach to a cell is if there is a perfect fit between a protein on its surface to the receptor. As I said, once those attach, it's actually the whole virus is taken into the cell. When we talk about some treatment options, one of the big treatment options is to actually get something that can attach to that spike protein so it can't attach to the cell. This is a great three-dimensional picture of that connection of the spike protein that you see here in red on the surface of the SARS virus to the ACE2 receptor right here. And this has to be a perfect fit, like two puzzle pieces fitting together. The reason that these coronaviruses are able to hop from animal species to animal species is because this particular receptor protein is very similar in different species. That's why it doesn't take much of an evolutionary change in that spike protein to have the ability to connect to this receptor. So once these two are connected together, the whole virus is taken into the cell. And Dr. Clifford's going to talk a little bit about what happens next here.
One of the pieces that makes this virus so interesting is both how it enters the cell and how quickly it can replicate itself. So like Ms. McBeath said, usually viruses that have RNA as their genetic material rely on some set of enzymes to incorporate that RNA into a DNA copy that's inserted into the nuclear genome of the host cell. But this cell gets right to work. It immediately takes a positive RNA strand and makes new versions of that strand that are replicated inside the cytoplasm. So now we have messenger RNA essentially floating around at very high concentrations in the host cell. This messenger RNA is immediately assembled into virus-specific proteins. And you can see in this diagram that there are endoplasmic reticular and Golgi apparatus vesicles that are involved in packaging up this virus neatly and in a very fast, organized manner to make new copies of the virus to be sent out of the plasma membrane of a host cell. So one of the issues that has really come up with this virus is that it not only replicates very quickly, but that it kills host cells fast enough that if these virus particles get down into lung tissue, they can actually interfere with the ability of the lung to deliver oxygen to the bloodstream because the virus particles themselves are killing so many host cells just by this very rapid infection mechanism. Now, there are a variety of ways to ameliorate what the virus can do. That is, sort of, instead of interrupt the virus's activity to sort of treat the symptoms or to try and reduce the virus's ability to pass through this cycle. And one of them are things like ACE inhibitors, which the only reason I've ever heard of ACE inhibitors are that my parents are all on them, and I hear about side effects of drugs on TV commercials, that if you're taking ACE inhibitors, they interfere with a lot of drugs. But one of the ways of lowering blood pressure is to reduce the number of receptors you make for that angiotensin converting enzyme. If you lower those receptors, you lower the amount of angiotensin, you lower the molecules that make your blood pressure rise. So that's one method of sort of providing kind of palliative or ameliorative care. Anti-inflammatory medications seem to be working. There was a big debate at the beginning about whether things like ibuprofen or aspirin were good treatments. But one of the effects that can happen later on is that your own immune system can start to overreact to not only the presence of the virus, but to the tissue damage that the virus itself causes. So when tissues get damaged, your immune system releases molecules called cytokines. And those cytokines are what tell local blood vessels to dilate and to start this whole redness, swelling, pain that we all associate with an inflammatory response. So if you add a viral attack plus an inflammatory response together, you might get a combinatorial effect downstream. And so that's one of the other things that can help make this virus particularly dangerous, that it works really quickly within a cell's own mechanism. It destroys tissue, and that it can turn a person's own immune system against them in an inflammatory response without an antiviral response. And so I'll turn over to Dr. O'Connor some of the sort of epidemiological and contagious parts of the virus that we've been able to discern, even in the short time that we've known about it.
Okay, so I liked this image, even though this is a model that has been going around. You might have seen it. It's been on a lot of the Facebook posts. It hasn't yet been tied to a peer-reviewed study at this point, but it's still a good graphical representation about the 6 feet distance that we keep talking about, and how that is different depending on what type of activity you may be engaged in. So I really like this visual because of that. We know that the virus is being transferred, as we just discussed, through water droplets. So that can include as you're running or walking. It's even if you're not actively sick and coughing, you're still, even if you know it or not, even as I'm talking right now, you have particles that are being emitted as you open your mouth, as you sneeze, you cough, etc. And those will remain in the air for a period of time and be transferred. That's why now we have the recommendation, even if you are symptom-free, that you cover your mouth with a mask when you're out in specific areas, overly crowded areas. What was interesting about this particular representation was trying to describe why it's also very important to think a little bit differently when you're not just walking, but perhaps running. Because the particles, as you're moving, you have to factor in your speed. They're becoming aerosolized and getting out into that air quickly. And the person is coming up behind you even more quickly. So the spread of the particles is going to be more broad, just based on the physics of the movement and how long they're moving. So the point of this particular article was not only keeping in mind getting to that 6 feet distance, putting a physical barrier, but also if you are doing a more rapid pace, or you're like running, for instance, making sure that your 6 feet is a circular distance, not just 6 feet behind someone, but also being to the left or the right of them, just in terms of the way the spray can be distributed.
And this contagious nature comes from, not only is it aerosolized, which Dr. O'Connor just talked about, but also very efficient attachment to the cell. This is a great attachment process here. Sometimes we call that the stickiness of the virus. So by the time you combine very efficient attachment, quick turnaround of making the viruses, and then the aerosolized nature, those three things combined is why we're at home right now, right? Because it's just those three make for the perfect storm for this virus to be transmitted. Okay, now we're going to turn into a little bit of the treatment options. You've been hearing a lot about this through the news and all of that. As Dr. Clifford said, the point is to break this cycle of the viral infection, and there's all kinds of different ways to do this, right? The first is to block the ability of the virus to attach to the cell. Lots and lots of work being done with what's termed monoclonal antibodies. These are antibodies that are actually produced in the laboratory to attach to the spike protein. By attaching to the spike protein, the virus can't attach to the cell. I personally feel this is what's going to, in the race to get a treatment, this one may win out. It's very similar to ZMapp for Ebola. Dr. Clifford, Dr. O'Connor, what do you think about the monoclonal antibodies?
One of the things that's really interesting about it is that we have to find a patient population that has recovered from the virus in order to recover these antibodies first. And so,
well, this is not the convalescent antibodies. These are the ones that are produced, and they actually have these from SARS-1.
Right. So we have to figure out sort of how antibodies can be collected in a useful way, and how specific they might be to this virus. I mean, I think the way the way to interrupt how the virus works is to find sort of the most catastrophic effect to the virus's cycle.
Mm-hmm.
And it seems to be that something that happens outside the cell is going to be a lot easier to facilitate than something that happens inside of the cell. So finding ways to prevent the blocking or to prevent a cell from moving from person to person are really the only two things we can do.
And the antivirals, when Dr. Clifford was talking about this process in the cell, one of the big ones are the treatment options that actually block the polymerase that you see right here. UH has actually been in a study of the remdesivir, which actually is a anti-polymerase treatment. So that shows some promise. And then Dr. Clifford, you talked a little bit about the intense cytokine storms, and there are some medications to try to block some of this inflammatory response and the treatment of acute respiratory distress. And then the passive immunity, where they actually take the convalescent antibodies from people who have already, they've been showing this stuff on, all the news channels of where people who have had the coronavirus and then recovered, they want their antibodies, right? And then the ultimate will be the vaccine, where we actually are given the antigen and then our own immune systems produce our self-antibodies and our memory cells, like getting a flu shot each year. Okay. Dr. O'Connor, anything to add here?
No, and I agree. Just getting back to the receptors, we also have to keep in mind, if you could go back to slide 6, please. One thing that we haven't talked about, though, in all cells, receptors have their own sort of life cycle, and more can be brought to the membrane as needed or pulled back inside the cell at different times. So finding a strategy that does inhibit that initial block, right, probably would be very effective initially, because as these receptors come in with the virus, the body is going to start producing more that can go back to the membrane, and we can keep having more and more interaction. So if you can stop right at the beginning, that really is a good target to initiate with.
Okay, and then lots has been in the news about origins of this virus, right? These are zoonotic, meaning that the coronaviruses do move from animal species to animal species. As I talked about, the receptor is very similar. We, as of now, have not found the exact viral genome of SARS-CoV-2 in any particular animal. There are very similar coronaviruses found in bats and also the scaly anteaters. But as Dr. Clifford said, this is a novel virus, right? It's brand new. So it probably evolved over time as different animal populations come in contact with each other and the human population. As we encroach on areas of the rainforest and things like that, this often happens. You hear a lot about the wet markets, right? The wet markets are really probably a site of amplification, whether, it actually evolved there or not, not so sure about that. But these are areas where humans come in contact with a lot of different animals and also their body fluid. So those are areas where, some amplification is definitely possible here. So who knows, right? Will we actually ever know? Probably not. But whenever you have different animal species coming in contact with each other, these abilities of these viruses to move is going to happen. So Dr. Clifford, Dr. O'Connor?
Yeah, I think one of the things to keep in mind that, for example, the flu virus, the biggest natural sink for the flu virus is in ducks. Mm-hmm. But humans don't spend all that much time interacting with ducks. So the way that a new flu strain gets into the human population is that it goes from ducks to pigs to humans. Mm-hmm. And so the biggest natural sink for coronaviruses is in bats.
Yeah.
But there are not that many situations where people are in close enough contact to acquire a respiratory virus from bats. And so we have to think about how the virus moved from a bat to something to people, in the same way that the flu very, very commonly does.
And that's the same thing with Ebola, HIV, all of these viruses have done species hopping and that kind of thing. So the last thing we want to talk a little bit about, Dr. O'Connor, with her great environmental aspect of this, lots going on here with the air pollution. So I'll turn it over to her.
Okay, thank you. And I'll go through these rather quickly because I want to make sure we have time for questions here. But just wanted to point out some of the positives, albeit these are likely, we'll find out just brief snippets of time once we get out of quarantine. These are likely to reverse fairly quickly. But the Himalayan mountains have been hidden from the people of India for decades due to massive brown air smog clouds that have been hiding over from all of the air pollution throughout the region. So it has been decades before the air has been actually clean enough for them to see the Himalayas. And obviously this is something beautiful, same with the canals in Venice. And I'm using these as examples that we're seeing, you know, clarity, and it's great, and we're seeing the animals come back, but this is more than just the beauty of seeing the Himalayas, right? These smog clouds create breathing problems, they create respiratory problems. So what's really interesting, and as this slide sees, just in a 1 month difference, okay, this is measuring nitric dioxide concentrations over China, and you can see the massive cloud in January, nearly completely gone, a few remnants here and there over some of the cities in February, but mostly gone after quarantine. And obviously, we know the virus is causing many deaths too, but even taking that into account, just the shutdown of these polluting factories and fossil fuel burning power plants is likely to have saved as many lives, up to 77,000 lives just in China. This is looking at the same readings over the United States, right? We're in a global environment, global weather patterns. So the clouds that we see do move across the entire surface of the earth. So we're seeing improvements in our own air quality too. So albeit short term, these are benefits. However, what I really want to talk about is what air pollution does and why it is actually a topic of conversation right now in terms of who is more susceptible to actually dying from the virus. So besides the gases that we know we're putting out, there's also actual soot or physical particulate matter that is being put out into the atmosphere. And these are measured based on their size. PM 2.5 is a particularly bad pollutant that is really bad in areas that are coal burning factories and power plants. And what happens is these particulate matters, they're actually solids that get up into the air, and they're small enough that when you breathe it in, if you're doing so at high enough quantities and over a long enough period of time, the defenses you have in your nasal cavity and your lungs, the little cilia that are supposed to help move all this out, get overwhelmed. And the particulate matter can actually bury itself into your lung tissue. Okay, so as Dr. Clifford was saying earlier, your lungs, when you're infected with the virus, right, you're not able to carry as much oxygen and other organ damage can occur. So now imagine that you have an individual who's been exposed, and all of us have really, to this PM 2.5, which on its own can cause some of the same symptoms that we do see with the virus itself. Now introduce the virus in these individuals who are already susceptible because their lungs aren't working at full capacity to begin with. And you can see why certain individuals might be more susceptible to longer term effects or even death from the virus. And unfortunately, what we're seeing is that there's a big inequality in terms of where this is happening, right? So we know if you live closer to the factories, if you live closer to the power plants, you more likely have higher exposure to these types of pollutants. You might not have as much access to healthcare, so you also have secondary effects such as cardiovascular disease and other respiratory issues, other problems that make you more susceptible and less likely to be able to fight off as strongly the symptoms of the virus.
Okay, Beth, so that concludes our presentation. If we have any other questions.
Yeah, we had one question come through on the chat, and please keep typing questions into the chat. The first one is from Shruti, asking, from what Ms. McBeath said, if we block the ACE2 receptor, does that stop the binding and therefore stop the entry?
Yes, definitely. As Dr. O'Connor said, if you could block the receptor, the virus isn't going to be able to get in. But remember, that receptor has a purpose for the cell, right? So going around and blocking all of these receptors may not be a good idea, right? But that's certainly an option. Probably the better would be to attach to the virus and have the spike protein not be able to attach to the receptor.
All right, we have a question from Emily. What makes a virus become seasonal?
This is a really important thing to think about with this virus in relation to things like common colds and the flu. You know, in colder temperatures, we're more likely to spend time indoors.
Mm-hmm.
The air that we surround ourselves with indoors in the winter is likely to be drier. So the mucous membranes of our nasal passages and our oral
have
passages
more of a chance to get in.
Yeah, are likely to be more fragile. So we can break those mucous membranes more easily. You combine that with being in close quarters with other people, and that allows a virus to spread more easily from person to person. If in the summer we're spending more time outdoors and have mucous membranes that are wetter, more intact, that it inhibits the virus from being able to replicate more quickly. It's not that those viruses go away. It's not that a virus disappears in the summer and reappears in the winter. What we're thinking about is how rapidly the virus can reproduce in an individual and how quickly it can spread from one person to another. And both of those things are easier when we are closer to each other in drier air. And so that's how seasonality of viruses comes about. We're not really sure whether this virus will be seasonal. This is, of course, the first season for the virus. And the fact that it's done so well in places in both the northern hemisphere and the southern hemisphere tends to suggest that it might not be as seasonal as things like the flu or the cold, but we don't have enough seasons yet to know whether that's going to be the case.
All right, we've got another great question coming from our students, from Shruti. What are your thoughts on herd immunity? I read a bit of how they tried it in Sweden. It doesn't seem to be working very well because their death rate is increasing, but experts still seem to be discussing it. And also, how would a potentially seasonal virus have an impact on the population with this herd immunity?
Well, herd immunity is always a good thing, as I'm sure Dr. Clifford and Dr. O'Connor would agree with. That's where you have a certain percentage of your population that are already immune to the virus, whether from a vaccine or having gotten it. And the issue, as Dr. Clifford mentioned, is this is a novel virus, which means it was brand new and nobody had immunity to it when it first reached the human population. So if we always rely on herd immunity really against all viruses, the question is, can we get that numbers high enough so that if somebody is not immune to it, that the herd immunity can protect them? And that's the issue, right? Dr. Clifford, would you agree?
Yeah, it's complicating that the herd immunity to this virus is almost zero.
Right.
And even for very deadly flu strains, they're strains of viruses similar enough that people have been exposed to before that the immune system can, even without stopping the spread of the virus, can slow the spread of the virus.
And make it not as severe.
Right, because it's sort of close to something that you may have seen before. This is not that. This is something that's totally new. And so herd immunity is going to be something that has to build over the course of potentially even decades.
Yeah, at least more. It's not going to happen in one season, right?
That's exactly right.
Yeah.
Right. We've got a question, nice segue, thinking about, from Mrs. Mueller, thinking about the vaccine and what your thoughts are on the chances, will it be free? What about testing? When are we going to see free, fast, readily available testing? Any thoughts that you have on those issues?
I think testing has increased on a daily basis. I think the new rapid tests that have come out are very significant. I don't think it will be very long where you're going to be able to go to your own doctor and get it similar to a rapid strep test. I don't think it's going to be very long, like a matter of probably a month or two for that kind of thing. It's coming pretty rapidly. I've been pretty impressed with, once those big companies got involved, I think it changed pretty rapidly in terms of testing. I think this vaccine will be the fastest to come to market. Dr. Clifford, Dr. O'Connor, what are your thoughts on that? On the vaccine?
Yeah, I absolutely agree. I mean, I think we've never seen a medical mobilization as fast as this in a global sense.
Mm-hmm.
You know, I think one of the things that makes it daunting is that we're constantly learning new things
Mm-hmm.
about this virus, and that's intimidating on the one hand, because you kind of have to keep checking the news, but also that there are so many eyes looking at this system that we're constantly getting new data, and we're even figuring out new ways to do the actual testing.
Yeah.
Mm-hmm.
You know,
Right.
whether it's a nasal swab or a saliva test, or whether it's detection of the RNA, whether it's detection of viral
The antibody.
proteins.
Yeah.
Right.
So
right.
it's
like
constantly changing, and the fact that it's changed so much for a virus that probably first made its jump to any person ever in October or November.
Yeah. It's been pretty quick.
Yeah, it's
been
really
very quick.
impressive, and you know, it might be that a virus is faster and cheaper than anything we've ever done before.
Okay, I am conscious of the time, and we're a few minutes over, so I think we will end here. I first of all want to say thank you so much to our biology teachers for that great presentation. Thank you to all of you for attending and coming up with such great questions. And if we didn't get to your question today, please feel free to reach out to any of us. We'd be happy to answer it. If you have a classmate or colleague that didn't make it to this forum, rest assured we are going to be posting the recording. And the final thought I would leave everybody with is, in a world of so much uncertainty, I think the one thing we can count on are brilliant scientists. And I think today's presentation was a great reminder of that. So thanks again, everybody. Have a great rest of your day.
Bye.
Bye-bye.
Bye.